Welcome. In this video, we are going to learn about agarose gel electrophoresis. It is used to segregate DNA, RNA, proteins, and other biomolecules. Today, we will see separation of PCR DNA samples based on their length. They are segregated by applying an electric field to move the charged molecules through the agarose gel matrix. The first step is to weigh the agar. After keeping the paper, we must bring the reading to zero in order to avoid considering the weight of the paper while weighing the powder. To prepare 80 ml of 0.8% agarose gel, powdered agarose required is around 0.64 grams. Then transferred to a conical flask. In order to prepare the gel tray for pouring, sellotape is put on both sides of it in order to create a seal around the tray so that it can hold the gel when it is in liquid form. The comb is then placed into the tray, which will help in creating the wells. TAE buffer of 0.5x concentration was prepared. TAE is a mixture of tris base, acetic acid, and EDTA. In the measuring cylinder, 80 ml of TAE was measured, which was then poured into the conical flask containing the agarose. The solution was then heated in the microwave for 30 seconds. This process may be needed to repeat again and again until the solution turns into a clear liquid. The solution is then let to cool to bearable temperature. Ethylene bromide, abbreviated as ETBr, is an intercalating agent commonly used as a fluorescent tag as it binds to DNA, which makes its binding pattern visible under UV light. ETBr is carcinogenic, hence gloves must be worn at all times while working with it. 3 microliters of it was added to the gel. Forty ml gel was poured into the tray and the rest 40 ml can be kept for later use by covering the conical flask with aluminium foil to avoid contamination. Buffer around 2 ml should be added to the gel before storing so that the gel does not lose much water content when reheated. The gel can later be reheated to liquefy and be cast again. Now to set up the gel electrophoresis apparatus, first we need the power supply unit. Before placing the gel in the tank, the sellotapes are removed from the sides of the tray to allow contact with buffer that will be poured later. The comb is then removed to expose the wells that were created. The TA buffer should be poured enough to completely submerge the gel. 5 microliters of the DNA sample is aspirated and placed on the sellotape. 5 microliters of the loading dye was aspirated and mixed with the DNA sample already placed on the sellotape. 5 microliters of 2x dye was added to 5 microliters of DNA solution to make it 1x. The dye and DNA must be mixed thoroughly before loading in the gel. Ten microliters of the solution was aspirated and loaded in a well. Glycerol in the loading dye increases the density so that the DNA sample nicely settles in the well. Otherwise, it will mix with the entire TAE buffer in the tank. Bromophenol blue in the dye moves in the same direction of DNA to help track the progress of electrophoresis visually. Three microliters of the 100 base pair DNA ladder was loaded in another well to be used as a reference. The lid of the tank was then closed properly and the power supply was switched on. The power supply was set to 100 volts and the gel was run for 30 minutes. After the fragments had gotten separated, the gel was then placed inside a gel lock machine without the tray. It helps us visualize the bands under UV radiation. The picture of the gel was taken using the camera present inside the machine, and its analysis can be done using the software of the gel documentation system. The shorter the fragment, the lower it is in the gel, and the larger the fragment, the higher the band will be in the gel.